Konnichiwa. Today, I will present you immunological research conducted in my laboratory. The title of this lecture is Microbial Symbiosis for Mucosal Fitness. As you may know, human gastrointestinal tract is colonized with 100 trillions bacteria, which estimate almost 10 times of the total cell numbers consisting of human body. Most of these commensal bacteria are beneficial and anatomically restricted either to the intestinal lumen or mucous layer of epithelial surface. Commensal bacteria colonized in intestinal tract contribute to the development of our mucosal immune system. For example, segmented filamentous bacteria belonging to gram-positive Firmicutes phyla, which strongly adhere to the surface of pile patches, enhance the differentiation of inflammatory cytokine IL-17 and IL-22 producing intestinal Th17 T helper subset. When these bacteria are orally fed into germ-free mice. This is another example relevant to the effect of commensal bacteria for the development of intestinal immunity. Oral feeding of mixture of Clostridium species isolated from human colon promote HOXP3 positive, CD4 positive regulatory T cell development in the colonic mucosa of the recipient mice. As I mentioned in the couple of previous slides, several commensal bacteria such as SFB and Clostridium colonized in our intestine exhibit beneficial effects for the development of our mucosal immune system. Furthermore, we notice these gut commensal bacteria contribute to the development of physiological function of the gastrointestinal tract, such as development of integrity of epithelial layer, development of neuroendocrine system, and development of intestinal vascular system. On the other hand, this biotic relationship, that is a disturbance of the homeostatic interaction of the intestinal microbes with the gastrointestinal tissue, deteriorates these important physiological functions and eventually develop pathological inflammatory conditions such as Crohn's disease, obesity, type 2 diabetes, and so on. As I mentioned in the couple of previous slides, several commensal bacteria such as SFB and Clostridium colonized in our intestine exhibit beneficial effects for the development of our mucosal immune system. Furthermore, we notice these gut commensal bacteria contribute to the development of physiological function of the gastrointestinal tract, such as development of integrity of epithelial layer, development of neuroendocrine system, and development of intestinal vascular system. On the other hand, this biotic relationship, that is a disturbance of the homeostatic interaction of the intestinal microbes with the gastrointestinal tissue, deteriorates these important physiological functions and eventually develop pathological inflammatory conditions such as Crohn's disease, obesity, type 2 diabetes, and so on. In general wisdom, gut commensal bacteria colonizing healthy mammals are anatomically restricted to either the intestinal lumen or mucous layer of the epithelia. Mucus, IgA, antimicrobial peptide, as well as epithelial integrity elegantly cooperate to segregate luminal commensal bacteria from direct interaction with intestinal epithelium to prevent microbial translocation to systemic organ and to avoid inflammatory diseases, suggesting that spatial segregation of commensal bacteria to luminal compartment is important for the maintenance of intestinal homeostasis. However, we recently discovered several environmental bacteria, such as bacteria species belonging to genus Alkaligenes, are resident within mucosal lymphoid tissue of intestine, such as pile patches, 1 decade ago. Bacteria species belonging to Alkaligenes contribute to mucosal IgA response and the development of type 3 innate lymphoid cell or the creation of immunological homeostatic condition in the small intestine. Further, we discovered gamma proteobacteria species, Stenotorohomonas species, living within tissue resident macrophages and contribute to the creation and maintenance of immunological homeostasis in the large intestine. So let me tell you how we explore this unique microbial symbiotic system within intestinal macrophages and their biological significance. At the beginning of this study, we explored whether microbes are present within tissue-resident macrophages. For this purpose, we performed 16S ribosomal RNA-based metagenome analysis by using genomic DNA purified from CD11b positive macrophages isolated from various intestinal tissues. As you can see in this slide, the microbiota composition analysis of pile patches CD11b positive macrophages showed predominant abundance of Firmicutes 57% of lactobacillus and 40% of segmented filamentous bacteria at the genus level, with trace abundance of Bacteroidetes 1.2% and Proteobacteria 0.7%. In comparison to pile patches-derived macrophages, chronic patchy-derived macrophages had higher abundance of Bacteroidetes 61% and Firmicutes 35%, but lower abundance of Proteobacteria 2.5%. On the other hand, non-lymphoid tissue chronic laminopropria CD11b positive macrophages demonstrated higher abundance of Proteobacteria 73%, followed by Bacteroidetes 21% and Firmicutes 3%. Stenotorohomonas species account for more than half, 52%, of the microbe within the chronic laminopropria macrophages. These results suggest Stenotorohomonas species are constitutively present within chronic laminopropria macrophages. We next conducted an in vitro assay to examine the morphological characteristics of Stenotorohomonas maltophilia colonized macrophages. For this purpose, we prepared bone marrow-derived macrophages and cultured them with the clinically isolated Stenotorohomonas maltophilia strain 13636 or environmentally isolated S. maltophilia strain 31559. As a control microbe, we use Escherichia coli strain K12. As you can see in this slide, of these three strains, only Stenotorohomonas maltophilia strain 13636 was able to enter bone marrow macrophages. In addition, transmission electron microscopic analysis confirmed the internalization of Stenotorohomonas maltophilia strain 13636 within both the endosomal and cytosolic compartments of bone marrow-derived macrophages. We next examined whether this persistent colonization of macrophages by Stenotorohomonas maltophilia creates mutually beneficial microenvironments within the colonized macrophages. In this regard, studies from the other group indicate that immunometabolism of tissue-resident macrophages is dramatically altered when pathogenic microbes are infected. To address this issue, we use a real-time extracellular flux analyzer to determine the oxygen consumption of Stenotorohomonas maltophilia colonized macrophages. As you can see in this slide, intracellular colonization by Stenotorohomonas maltophilia strain 13636 increase the spare mitochondrial oxygen consumption of bone marrow-derived macrophages when compared with that of control macrophages, suggesting that macrophage persistently colonized by Stenotorohomonas maltophilia human isolated strain are likely to show mitochondrial oxidative phosphorylation-dependent immunometabolism, similar to tissue-resolving type macrophage, such as alveolar macrophages. In addition, bone marrow-derived macrophages cultured with Stenotorhomonas maltophilia strain 13636 show robust IL-10 production. This is a typical characteristic of tissue-resolving M2 macrophages. In contrast, bone marrow-derived macrophages cultured with the environmentally isolated Stenotorhomonas maltophilia strain show only marginal IL-10 production. Interleukin-10, IL-10 is a typical anti-inflammatory cytokine which contributes to the creation and maintenance of immunologically homeostatic environment in the intestine. In addition, other studies strongly suggest chronic macrophages preferentially produce IL-10 when compared with other tissue-resident macrophages. Because we notice enhanced IL-10 production by bone marrow-derived macrophages colonized with clinically isolated Stenotorhomonas maltophilia, we next examine the role of IL-10 in the maintenance of cytosolic colonization of Stenotorhomonas maltophilia in macrophages. As you can see in this slide, clinically isolated Stenotorhomonas maltophilia strain was not able to colonize IL-10 deficient macrophages in comparison to IL-10 competent macrophages. In contrast, the addition of recombinant IL-10 to the IL-10 deficient macrophage effectively recover their intracellular colonization. Furthermore, exogenous IL-10 addition enhances the cytosolic inhabitation of Stenotorhomonas maltophilia in wild-type IL-10 competent bone marrow-derived macrophages. These findings suggest clinically isolated Stenotorhomonas maltophilia strain induced robust IL-10 production from host macrophages, which support the habitation of Stenotorhomonas maltophilia in the cytoplasm of macrophages. We next try to identify a symbiotic factor from Stenotorhomonas maltophilia strain 13636 that is responsible for their ability to live within macrophages. Because in general wisdom, microbe-derived symbiotic factor for persistent colonization within host cell are induced under nutritionally poor or acidic condition. We cultured Stenotorhomonas maltophilia strain in a chemically defined, nutritionally limited M9 medium that contain minimal amounts of glucose. We then concentrated this Stenotorhomonas maltophilia culture supernatants and separated through SDS page and revealing that the clinical isolate Stenotorhomonas maltophilia strain produced a 25 kilodalton protein that was absent from the products of environmentally isolated Stenotorhomonas maltophilia. Furthermore, Maldi-Toffmas analysis revealed that this 25 kilodalton protein corresponded to the hypothetical protein encoded by SMLT 2713 in S. maltophilia. To explore the effect of the hypothetical protein encoded by SMLT2713 on cytokine production, we express the gene in bone marrow-derived macrophages. As you can see in this slide, the expression of SMLT2713 induced the production of IL-10 in bone marrow-derived macrophage without effect on other cytokines such as IL-1 beta, IL-6, and TNF-alpha. To examine the role of SMLT2713 protein in the persistent intracellular colonization of macrophages by Stenotorhomonas maltophilia strain 13636, we next constructed SMLT2713 deficient Stenotorhomonas maltophilia mutants. As you can see in this slide, the constructed S. maltophilia SMLT2713 deficient strain was not able to establish persistent intracellular colonization in macrophages. The SMLT2713 deficient mutant strain instead aberrantly colonized endosomally vesicles and occasionally induce a morphology similar to pyroptotic cell death in the infected bone marrow-derived macrophages. To examine the physiological significance of the Stenotorhomonas maltophilia-derived SMLT2713 in vivo system, we adaptively transferred bone marrow-derived macrophages transduced with SMLT2713 gene to the CD45 RB high T cell induced enterocolitis model. As shown in this slide, histological analysis of the distal colon isolated from the CD45 RB high T cell reconstituted mice together with the SMLT2713 gene transfected macrophages markedly ameliorated the inflammatory lesion, such as disruption of epithelial layer and accumulation of mononuclear cells into the lamina propria, when compared with the control CD45 RB high T cell reconstituted mice. Finally, we assessed whether long-term persistent colonization of Stenotorhomonas maltophilia in colonic resident macrophages actually demonstrate the symbiotic habitation of the bacteria in macrophages by constructing Stenotorhomonas maltophilia colonized notobiota mice. When we constructed Stenotorhomonas maltophilia colonized notobiota mice by oral administration of Stenotorhomonas maltophilia into germ-free mice, the notobiotic mice colonized with SMLT2713 producing S. maltophilia strain exhibit slight increase of HOXP3 positive CD4 T cell in the colonic lamina propria when compared with germ-free mice and notobiotic mice colonized with SMLT2713 deficient Stenotorhomonas maltophilia mutant strain. From this in vivo experiment, we conclude the long-term persistent colonization of Stenotorhomonas maltophilia strain within macrophages contribute to the status of the long-term to the creation of immunologically homeostatic condition in the colon. Therefore, the status of the long-term colonization is defined as symbiotic cohabitation of S. maltophilia within the macrophage. Furthermore, this 25 kilodalton exported protein from S. maltophilia, termed as SMLT2713, actually act as a symbiotic factor for the host macrophages. I thank all of my collaborators very much for their devoted contribution to improve this study. Thank you for your kind attention.